It's 1:32, so we'll get started. So for those of you who don't know me, my name's Jessica Young. I'm an assistant professor in lab med and pathology, and I'm also part of the Institute for Stem Cell and Regenerative Medicine. And it is definitely my pleasure and honor to introduce Ashleigh, Dr. Ashleigh Schaffer, who I've known, gosh, almost 10 years, 9 or 10 years, as we were postdocs together at UC San Diego. And she's now in the Department of Genetics and Genome Sciences at Case Western Reserve University in Cleveland, right? Or Columbus, geez, I was even there, Cleveland, Ohio. But just to give you a brief background, Ashleigh received her PhD in biological sciences from the University of California at Irvine in 2010. And then she went on to do postdoctoral training, really focusing on genetics at UCSD, working in the labs of both Dr. Joe Gleason and Jean Yo. And during this time, she did really innovative work on mutations in RNA-binding proteins that lead to developmental brain malformations, and she developed stem cell models for these disorders. And her work has been really, she's published in really high profile journals, just to name a few: Cell Development, Neuron, Nature Genetics. That's an awesome scale of work. She was also the recipient of the NIH Pathway to Independence Award, the K99R00 Award, and that's how she started her own lab at Case in 2017. And notably there, she's been appointed as a prestigious Mount Sinai Scholar, which is a program that supports the medical school's most promising young faculty in basic sciences. So I'm very happy to have her present today. And I believe she's going to talk a little bit about a new direction in her lab, which is the role of the RNA kinase CLIP1 in both motor neuron disease and how it can contribute to broader implications in neurodegeneration. So we'll welcome Ashleigh's talk, and I believe the way this will work is at the end of her talk, you can type your questions into the chat, and I will do my best to get moderate and make sure they all get answered. So thank you, Ashleigh.
Hey, let me just start by sharing. Is it working?
I think you're still in like presenter mode where we can see your notes slides at the bottom.
Oh, that's weird. Okay, here.
Uh-oh. Ashleigh, did we lose you?
I'm sorry, I'm back. Let me try again.
That looks great.
That's better. Okay. Sorry there. Okay, so first I want to thank Jessica for her kind introduction, and this opportunity, like she said, is a continuation of some of my postdoc work into how we established the lab at Case and kind of what our main project has been over the past 3 years. So thank you again for the invitation. It's very nice to see everybody and talk to you, even if it's one day before Thanksgiving. So let's see. Okay, so our lab is mainly interested in understanding how human brain develops and how this is maintained. And defects in these processes often lead to neurological disorders that generally have no cure or treatment. So there's a great need to find answers to these questions. And the approach that we take is to use human genetics as a screening tool to identify genes and pathways that regulate these processes. And in doing so, we can also help to inform the genetic community about variant pathogenicity, which is a huge bottleneck to treatment development as well as precision medicine. So interestingly, human genetics research has shown that mutations in essential genes, or genes that when deleted cause lethality, disproportionately cause unique orphan diseases, many times with neurological involvement. So the concept that a ubiquitous cellular process can affect some cell types and spare others is known as selective vulnerability. And the causes of selective vulnerability are unknown and also a current focus of our research. So to find new disease genes or investigate the causes of them, we follow this general flowchart. We start by recruiting patients with an interesting phenotype and then perform genome sequencing to find variants. If the patients have a mutation in a known gene that would explain their phenotype, then they proceed to genetic counseling to obtain guidance for their care and family planning. If they have a variant in a new gene or a gene that has not been previously linked to their disease, then we need to do further testing to show that this mutation could be causative. And this is where the work in our lab comes in, which is to develop disease models and functional tests for the human mutation. In addition to this, if we have additional families or multiple families that all have mutations in the same gene, we can start to determine genotype-phenotype correlations that can help point to the affected pathways and potentially precision therapies for these patients. We can also work with our collaborating physicians to clinically define new syndromes, and this aids future patients in getting a more accurate diagnosis, as well as helps us limit our recruitment efforts to only new diseases. So we've been focusing mainly on recessive diseases because loss of function variants account for up to 70% of the orphan diseases that I showed you earlier in the Venn diagram. So to help improve our chances of finding new disease genes, we recruit patients primarily from the Middle East. This area of the world has high rates of marriages between first cousins. So in this small pedigree shown on the right, if a founder mutation is present in one of the grandparents and passed down to the parents as heterozygous, so when the parents have children, the mutation would have a 1 in 4 chance of being present in a homozygous state. So when the mutation is pathogenic or loss of function, this would lead to these kids having a disease. And if it's heterozygous or normal, then individuals would be healthy. So we collect the genomic DNA from all the available family members in the pedigree and perform genome sequencing. We then align the sequencing reads and determine areas in the genome that are uniquely homozygous in all the affected individuals. And this is called homozygosity mapping. And the reason we do this is because the pathogenic variants are more likely to exist as homozygous if the mutation is the same and came from one of the grandparents. We then look within these homozygous intervals for mutations that are predicted to be damaging by variant annotation software, such as like Polyphen or SIFT or CAD. We then look at where the gene is expressed and what it's predicted to do. So for instance, if you're looking for a primary microcephaly gene, you might look for genes that regulate the cell cycle or control microtubule stability. And this could lead you to prioritize those potential mutations over others. So if other patients have mutations in the same gene, that's also a hint that this could be potentially new disease-causing gene, especially if those patients have the same clinical presentation or phenotype. So after seeing all the potential candidate genes, we prioritize them and test their inheritance pattern by Sanger sequencing. And then we pick our favorites to bring forward into the lab for functional analysis. So here are some examples of disease pathways in neurological disease. I just put up 2: autism spectrum disorder, which is a childhood onset, potentially neurodevelopmental or postnatal disease, and as well as ALS, which is an adult onset motor neuron disease I'm sure many of you are familiar with. And you can see that processes that are thought to be essential for every cell type can lead to these different diseases. So for instance, in autism spectrum diseases, you see chromatin organization, histone modifications, RNA processing and metabolism, Whereas in ALS, you see mitochondrial function, endosome trafficking, chromatin remodeling, oxidative stress. So you may have noticed that RNA processing and chromatin organization were listed in both of these diseases, but the onset of the disorders and the cell types that are affected in these two diseases are completely different. So we're interested in why that might be. So one possible explanation is that there are over 400 cell types in the human body that all have the same DNA, but unique gene expression patterns. And our lab specifically focuses on neurons, which are highly specialized cells of the central nervous system or nervous system with multiple unique features. They are polarized, have cell bodies and dendrites, as well as single axons. And most of them are born during embryonic development and need to be maintained throughout life. There are also different neuronal subtypes. So with single cell RNA sequencing, we now know that there are over 200 distinct subtypes of neurons. And here's an example of a Golgi stain of a newborn brain showing differences in size, structure, and projections within the cortex. Neurons are also the largest cell type in your body. So from this whole body prep of Harriet Cole, which was performed in the 1880s, you can appreciate how long some of the motor neurons are. They extend from the lumbar region of the spinal cord all the way to the tips of the toes. Another large neuronal cell type that is vulnerable in many neurological diseases is the Purkinje cells of the cerebellum. And they have a distinct dendritic arbor and a characteristic large cell body. So today, as an example of our work, I'm going to focus on a rare childhood neurological disorder called pontocerebellar hypoplasia. And this disease is characterized by progressive atrophy of the pons, brainstem, and cerebellum as shown in this image. There are no treatments available to patients, and the cause of the disease is unknown. Most patients that have pontocerebellar hypoplasia have mutations in the gene TSEN54, although there are many other subtypes. So these subtypes are defined by clinical features as well as by more recently mutations in other genes. So most of these causative genes are involved in RNA processing and metabolism. So many people speculate that this disease is thought to result from improper RNA biogenesis. So from a large cohort of patients from the Middle East, when I was a postdoc, we identified 4 families that had similar brain features that were consistent with pontocerebellar hypoplasia. We performed genome sequencing, like I mentioned, and found a rare missense mutation in arginine to histidine at position 140 in the gene CLIP1. This mutation appeared to be a founder mutation because it was only identified in individuals of Turkish descent. And through looking at recombination, we were able to identify or project that this mutation was originated during the Ottoman Empire period in Turkey. So the variant followed a strict recessive mode of inheritance, and all the carriers reported no neurological symptoms. This mutation occurs near the P-loop or the RNA kinase domain of the protein and is highly conserved down to yeast. So as shown in the cartoon on the right of tRNA processing, CLIP1 associates with the TSEN proteins in step 2. tRNAs are short, non-coding RNAs that are essential for protein synthesis. They are thought to be highly redundant because there are over 500 genes encoded in the human genome, many which recognize the same codon sequence and carry the same amino acid. So here's a closer look at this tRNA splicing pathway. So there are 2 potential ligation pathways following the cleavage of the tRNA intron by the TSEN proteins. So if CLIP1 is used, the 5' hydroxyl group on the 3' exon is phosphorylated, and that leads to ligation by a yet to be unidentified ligase. But if CLIP1 phosphorylates this 5' hydroxyl group, HSPC117 is unable to ligate these 2 exon halves to form a mature tRNA. So these 2 pathways are thought to be mutually exclusive. Both of them have been identified in human cells, but whether all cell types in the human body use both pathways or some cell types just use one of the pathways is currently unknown. So to determine how this R140H missense mutation in CLIP1 might affect its function, we first modeled it on the partially crystallized structure of yeast CLIP1. And we found that the R140H mutation disrupted a conserved inferred hydrogen bond. So to determine if this structural change affected protein stability, we were able to obtain primary fibroblast cells from patients with homozygous mutations in CLIP1, as well as the unaffected controls. And we performed a Western blot for CLIP1 and found that there was no change in expression. So to us, this indicated that the mutation was likely not destabilizing the protein and could be affecting its function. So since the mutation occurred near the kinase domain, we wanted to next test kinase activity. So I produced a recombinant GST tagged CLIP1 and tested its kinase activity using a polyadenylated RNA. We used the T4PNK as a positive control for the assay, and we found that the kinase activity of the CLIP1 R140H mutation was reduced by 60% compared to control. And this indicated to us that, that this mutation is likely hypomorphic and that it has reduced function compared to controls. So now we wanted to understand how this change in protein function might alter tRNA processing. So we decided to use an induced neuron protocol, which directly converts fibroblasts into neuronal cells. Roughly 70 to 90% of the fibroblasts, or I guess they're induced neurons, will be TUJ1 MAP2 co-positive within the first 7 to 10 days. We can then use these cells to look at cell type specific tRNA splicing defects. So since CLIP1 is predicted to function in tRNA splicing, I first performed a northern blot for intron containing tRNAs, isolated from the patient fibroblasts as well as the induced neurons. We found tissue-specific increases in certain intron-containing tRNAs, suggesting that certain tRNAs are differentially expressed across tissues and may be tissue-specific. We also found an increase in intron-containing pre-tRNAs and a decrease in the mature tRNAs, specifically in patient-induced neurons, but not in the fibroblasts from patients. So this suggested to us that tRNA processing might be specifically impacted in neurons, and that could explain why the patients have neuron specific or neurological phenotypes, but not phenotypes in other tissues. So we next wanted to address this question, and we, looking back on past literature, one of the common defects that lead to pre-tRNA accumulation in yeast was the mislocalization of the endonuclease complex. So we performed immunostaining for CLIP1 in the patient cells and found that the mutant CLIP1 was being mislocalized to the cytoplasm, specifically in the patient cells compared to controls. We observed this defect in both patient fibroblasts to a small degree, but it was a much stronger phenotype in the induced neurons. And this might explain why we were unable to detect changes in tRNA splicing in the fibroblasts, but saw such a strong molecular phenotype in the induced neurons. So if this mutation leads to CLIP1 mislocalization, how might that affect the function of the tRNA splicing endonuclease complex? So through a collaboration with Chris Trotta at PTC Therapeutics, we wanted to test the interaction between wild type and mutant CLIP1 by transfecting a flag his tagged CLIP1 vector into 293H cells. We then double affinity purified the CLIP1 bound protein complexes and looked for the presence of the TSEN complex proteins. TSEN54 was used as a positive control, and we found that the patient mutation showed reduced affinity for TSEN2 and TSEN34, and these are the two catalytic subunits of the tRNA splicing endonuclease complex compared to the wild type CLIP1. So now we know that the patient mutation alters interaction with its canonical binding partners. And we wanted to test how this could change tRNA cleavage. So we tested the purified complexes in an in vitro model of tRNA cleavage. In this model, you add exogenous pre-tRNAs to the CLIP1 purified proteins and then analyze cleavage of the intron at 15 minutes, 30 minutes, or 60 minutes by this blot, radioactive blot. So the yeast tRNA endonuclease is used as a positive control, and you can see that the complexes containing the patient mutation were unable to cleave the pre-tRNAs, while the wild type CLIP1 bound complexes had a robust cleavage activity. So these results suggested that the CLIP1 R140H might be impairing tRNA biogenesis by affecting the splicing of the pre-tRNAs, specifically within the neurons. So this was really exciting to potentially uncover the pathogenic mechanism of a disease for pontocerebellar hypoplasia. But we were really intrigued by the differences in the clinical presentation of patients with, or PCH patients with variants in TSEN proteins compared to CLIP1. So I want to draw your attention to the underlying text, which describes that the CLIP1 patients have a very severe motor neuron disease, while the patients with TSEN variants do not. So we hypothesized that the motor neuron disease might be caused by a tRNA independent mechanism, or potentially even a cell type specific mechanism. So to investigate this, we reprogrammed patient fibroblasts into induced pluripotent stem cells and differentiated them to motor neurons in vitro. And we found that the motor neurons differentiated with very similar efficiency and identity. However, during the motor neuron maturation process, we found that there was a reduction in cell density, specifically in cells that came from the pontocerebellar hypoplasia type 10 patients. So we next wanted to test whether this phenotype was due to the tRNA processing defects. So we sequenced the mature tRNAs and performed northern blots like I showed previously. And unlike what we saw in the induced neurons, we found that the motor neurons had similar expression and splicing of the tRNAs. So this suggested that tRNA processing was not affected in the motor neurons, and that another mechanism was potentially at play leading to this phenotype that we saw in the dish. So in addition to tRNA processing, CLIP1 is a core component of the mRNA cleavage and polyadenylation complex, which is this cartoon I'm showing here. It's part of a very small subunit called CF2M, which is depicted in green. And when we examined the association of CLIP1 with the components of this complex by co-IP, we found that the patient mutation impacted association to its direct interacting partner PCF11, as well as the complex as a whole, shown by its loss of interaction with simplekin. So following transcription, pre-tRNAs or pre-mRNAs also go through multiple co-transcriptional processing steps including the splicing of introns and the cleavage at the 3' end. So the cleavage and polyadenylation complex is shown in purple on this slide, and it recognizes a specific motif in the transcript called poly-A sites. The co-transcriptional mRNA processing all happens together at the same time, so this results in different combinations of mRNA isoforms. Changes in splicing can lead to skipped exons, alternatively used splice sites, mutually exclusive exons and retained introns. An alternative transcription initiation leads to alternative first exons, and alternative cleavage and polyadenylation leads to intronic polyadenylation, which in this slide is called alternative last exon, as well as the tandem 3' UTRs, where the length of the canonical 3' UTR changes. So overall, these alternative processing events, they change the mRNA isoforms that are expressed, and they can affect the coding potential, the transcript stability, as well as the localization of these transcripts within neurons or cells. So for the rest of this talk, I'm going to focus specifically on these last two, which are caused by alternative cleavage and polyadenylation events. So to identify changes in splicing and gene expression, we used RNA sequencing. This is a pretty standard protocol. We used ribosomal RNA depletion, and then all of the non-coding and mRNAs are amplified and sequenced to determine their expression level. And to detect the 3' ends, we used an approach called poly-A-click-seq. And in this case, you use a poly-T primer to amplify just the last 250 nucleotides of a transcript. So after sequencing, we can pick up peaks of reads, which are equivalent to how many transcripts each poly-A site, how many transcripts used each poly-A site. So for instance, the black track on the top uses the first or the proximal poly-A site, whereas the red track on the bottom uses the last or the distal poly-A site. So here are the results from the poly-A-click-seq when we compared the patient lines to the controls. So each dot is an mRNA isoform. The red dots are mRNA isoforms that are different between the affected patient motor neurons and the controls. And this is plotted by fold change. On the right, we took all of the red dots and determined where in the gene body they would be located. We found that the affected patient motor neurons showed less poly-A sites in the intron and more poly-A sites at the canonical 3' UTR. This suggests that the patient mutation suppresses selection of these intronic polyadenylation sites. This reduces the number of short mRNA isoforms that are being expressed and increases the number of long mRNA isoforms that are expressed. This effect was also observed when we looked at splicing and gene expression of the mRNA isoforms by total RNA-seq. So in the top right graph, we plotted whether exons were included in yellow or excluded in blue and their position across the gene. The TSS is the transcription start site and the TTS is the termination site. We found a net increase of exons at the 3' ends compared to the decrease in exons, indicating that the genes were getting longer overall. This effect was specific to genes that had poly-A sites in their introns. So in the violin plot on the bottom right, we compared the gene length to expression levels. So only long genes that had intronic polyadenylation sites showed an increase in expression. This is a little easier to see in the scatter plot on the right, where the gene length and fold change are shown. So the color of the dots represent how significant the change is, and the average length of a human gene is shown by the black line near the bottom. So you can see that most of the upregulated genes are very, very, very large. To understand how these transcriptional changes might impact neuronal function and survival, we performed GO-term enrichment analysis for the differentially expressed genes. So the genes that encode multipass ion channels and synapse-associated proteins were predominantly found in the upregulated groups, and the short genes that control DNA binding and damage were downregulated, indicating that the patient mutation likely disrupts ion homeostasis and neuronal activity. We have some preliminary data from a multi-electrode array in the lab showing that it leads to a decrease or a hypoexcitability phenotype in culture. So up to this point, all of the transcriptional analysis was performed on patient-derived cell lines, and we wanted to see if we could validate our findings in a physiological disease model. So we used CRISPR-Cas9 to introduce the patient mutation in mice and then analyzed their neuromuscular junctions and motor function. So the NMJs are marked by the presence of the presynaptic marker synaptophysin, which is shown in green, which is present in the neuron, and the postsynaptic marker, which is alpha-bungarotoxin, which is located on the muscle side. So overall, we found a similar innervation pattern between the mutant and control mice. However, the size of the neuromuscular junctions was reduced by about 30% by 1 month of age. So to test whether this reduction in neuromuscular junction size affects motor function, we performed a stride length test. So in this test, you paint the back feet of the mice with ink, and then you let them walk on white paper. And then you can measure the distance of their stride, with smaller strides correlating to impaired motor function. So over time, what we observed was a decrease in stride length in the mutant mice compared to the control mice, indicating that they had reduced motor function. So since the mice had a shared motor neuron phenotype with our PCH10 patients, we next wanted to test whether we could detect some of the same transcriptional changes that we found in patient-derived motor neuron cultures. So we crossed our mutant mice with a motor neuron reporter line, so we could isolate primary motor neurons by FACS. We then performed qRT-PCR for the differentially expressed genes in RNA isolated from these motor neurons, as well as the patient-derived motor neurons, using the same primers. Of the 6 that we tested, 5 of them showed a similar expression pattern. This one exception was alpha-synuclein, which was not overexpressed in the mouse embryos, but was overexpressed in the patient iPS cell-derived motor neurons. This indicated that the patient mutation likely also disrupts mRNA processing in vivo, but the gene sets might be slightly different. So this led to our model of what was happening in pontocerebellar hypoplasia type 10. So we believe that CLIP1 participates in intron-containing tRNA splicing in the brain, or it's essential for this process in the brain. And this would be supported by our functional work with our induced neurons, as well as the clinical presentation of patients with variants in the TSEN proteins, as well as CLIP1. But in motor neurons, we think that tRNA processing could be compensated by the RTCB or the HSPC117 pathway, or that CLIP1 is not necessary for TSEN function in that cell type. Instead, we think that CLIP1 normally associates with the cleavage and polyadenylation complex, and that loss of this association can lead to a suppression of intronic polyadenylation and an upregulation of long mRNA isoforms that have these intronic polyadenylation sites. And these genes just tend to disproportionately encode for proteins that are important for neuronal function because they're long, and neurons express longer genes with more mRNA isoforms than other cell types. These genes are sometimes also expressed specifically in neurons, so this might explain why the motor neurons are affected, whereas skin or liver cell types would not be affected. So this could explain why a single variant, such as what we found in CLIP1, could lead to cell type specific phenotypes, even though it is a ubiquitously expressed gene and a ubiquitous protein. So in the future, we hope to dig further into these molecular changes to determine if these neurodegenerative disease mechanisms are shared with other disorders. So I'm just going to give you a couple of interesting examples that we're just beginning to investigate. So our first step into this was to determine whether this unique transcriptional signature of intronic polyadenylation evasion was found in other neurodegenerative diseases. So we surveyed all the other pre-existing 3' end mRNA sequencing data sets, which unfortunately there isn't very many, like a total of 6. But in this study from 2012, Rin and colleagues identified an APA event in alpha-synuclein gene that led to increased expression of a long 3' UTR that had a single nucleotide variant that would increase the expression and localization of the protein. And this was associated with pathology of the mitochondria. So since there was a precedence for increased expression of long mRNA isoforms in Parkinson's disease, we performed our IPA analysis and found that intronic polyadenylation was reduced in PD. So similar to CLIP1 R140H patient-derived motor neurons, we saw that the long genes with intronic polyadenylation sites had a statistically significant increase in expression. And this highlights a potentially shared novel mechanism of gene regulation that could be occurring in Parkinson's, sporadic Parkinson's disease, as well as potentially other neurodegenerative diseases. But the mechanism controlling these 3' end processing changes described in Rin's paper have not been discovered, and CLIP1 has not yet been associated with Parkinson's disease. So this mechanism at this point is still unclear for Parkinson's disease. But in contrast, recent genetic mediation analysis using GWAS data from 25,000 Alzheimer's disease patients and 48,000 controls, as well as brain gene expression from 350 individuals from the Religious Orders Study and Memory and Aging Project, found 206 new causal target genes for Alzheimer's disease, and CLIP1 was number 3 of the most robust target, explaining roughly 11% of the variants, suggesting that upregulation of CLIP1 contributes to Alzheimer's disease. So indeed, a recent paper recently investigated transcriptional dysregulation in brain tissue from a cohort of 36 individuals with Alzheimer's disease. So they used an alternative mRNA 3' end sequencing method similar to what we did, and they found that the pattern of mRNA 3' UTR expression or alternative polyadenylation site usage predicted Alzheimer's disease better than total gene expression or even pathology alone. And this was specifically associated with an increased expression of 2 of the 6 altered RNA-binding proteins shown in J, TDP43 and CLIP1. So TDP43 phosphorylation and aggregation is well studied, and it's been linked to many neurodegenerative diseases like ALS and frontotemporal dementia. And altered TDP43 function has been shown to lead to disruption of multiple cellular processes and impaired cognition. This is just an example of some brain tissue from patients with AD or pre-AD or non-AD, and you can see that the phosphorylated TDP43 aggregates in the cytoplasm, whereas normally it's supposed to be found within the nucleus. So what does CLIP1 look like in Alzheimer's disease brain tissue? So we were able to obtain hippocampus tissue sections from our Cleveland Alzheimer's Disease Research Center from patients with different stages of Alzheimer's disease and controls. And we stained the slides for CLIP1 with a counterstain of hematoxylin. And we found that CLIP1 was specifically highly expressed in patients with AD, but not the mild or moderate AD patients. And we also found that CLIP1 was mislocalized to the cytoplasm in many of the neurons and showed very high levels of expression in the neuropil as well as in the cell bodies. But how this change in CLIP1 expression affects its normal function in tRNA processing and mRNA processing is unknown, and whether these changes contribute to disease is a big question that we want to address in the future moving forward with the help of collaborators. And the reason that we're interested in this is because we find that there are a lot of parallels between what we observed in AD patient tissue and what we saw in patients with the CLIP1R140H, specifically the reduction in nuclear expression of CLIP1 and an accumulation in cytoplasmic aggregation. So with that, I just want to summarize what I've described to you so far. So our research focuses on pediatric neurodevelopmental disorders and neurodegenerative diseases, and we use human genetics as a tool to uncover the function of essential genes in these disease types. Our recent work on the essential gene CLIP1 identified tissue-specific requirements which may partly explain the clinical presentation of the patients. And I focused on a rare pediatric neurological disorder called pontocerebellar hypoplasia, where the majority of variants are found in tRNA splicing proteins, including the T-cell proteins and CLIP1. And the CLIP1 variants led to a very unique subtype of pontocerebellar hypoplasia, where the patients had motor neuron disease. And through our functional analysis using patient-derived cells and mice, we're able to show that this variant impacts tRNA splicing in some cell types, but not others. In motor neurons, the patient mutation altered mRNA isoform expression by suppressing expression of short isoforms through alternative polyvinylation. And using mice as a disease model, we are able to confirm some of these transcriptional changes in vivo, indicating that motor neuron disease is likely caused by a tRNA independent mechanism. The transcriptional mechanisms that we uncovered in this rare disease were also found in Parkinson's disease, and patients with AD had increased mislocalized expression of the CLIP1 protein, similar to what we observe in our induced neurons that have the CLIP1R140H mutation. We have yet to determine whether misregulation of CLIP1 contributes to these more common adult onset neurodegenerative diseases, but it lends support to the idea of using a rare disease genetics to learn more about human brain development as well as brain health. And lastly, I want to acknowledge all the individuals and groups who contributed to this work, especially the patients and their families. This work would not have been possible without excellent collaborators, including my former mentor, Joe Gleason, and my current collaborators at UTMB and Case Western, as well as our funding sources. And with that, I would be happy to take any questions that you might have.
Great. Thank you, Ashleigh. That was a really great talk. So I see some questions showing up in the chat. So I'll go ahead and start there. Wait, Ashleigh, are you still there?
Yes.
Okay, good. Sorry, you went off my Zoom screen for a second.
Sorry.
Okay, so yeah, I'll start with moderating some of these questions in the chat and we'll see how far we get. So Ellen Weissman is asking, related, I think, to your sequencing data, whether the tests of expression being run, are they being run as if expression of every gene is independent of the other genes? Because there may be a correlation in your data because of multiple RNAs measured from the same subject or cell.
Let's see. So for the GO-term enrichment analysis, we definitely took into consideration only the genes that were expressed in motor neurons to identify enrichment of processes. For the general gene expression analysis, like the RNA-seq and the PAC-seq, they were independently counted and compared by across genotypes. I don't know if that helps to answer that question.
Ellen, are you there? Do you want to jump in and say if that, oh, but you're muted.
Wrong button. Sorry, we'll do it this way. I missed the first half because I couldn't get in. So I sort of walked in and tried to follow things. So, you know, the problem is when you do have multiple measurements from an individual, they're probably correlated. And so I worry sometimes when I see those kinds of tests which are being carried out as if they're independent. I think that's one of the reasons we have so much trouble replicating studies is the tests are not taking that correlation into account. So I think that's technically not correct to do it that way. Let's put it that way.
For the PAC-seq data, we didn't compare like absolute levels, but it was the ratio of usage of the poly-A sites.
Right, that's even worse because ratios are bad.
Well, the problem is that when you use one site, you automatically use less of the other site, right?
Yeah,
so
right. Yeah, exactly. I understand. I understand that. I understand that. I'm just commenting that there are problems to doing the analysis that way, and you sort of lead yourself astray sometimes when you do that. Talk to your statistical colleagues, and maybe they can help you out with the analysis. It's not something for somebody who doesn't have the right training to do, but there are better ways of doing that analysis, and maybe you'll have better, more followable results in the end. Is the issue. You don't want to be going down a rabbit hole.
Right.
Right. That's not a good thing to do. So.
Great. Thanks. Okay, Eleanor is asking, does the CLIP1 knockout recapitulate CLIP1 PR140H phenotypes?
No, so in the mice, it's embryonic lethal, and in the stem cells, they're actually opposite. So what we found is that the knockout led to increased intronic polyadenylation. It would be consistent with like de-repression of its binding partner PCF11, and that the R140H had a dominant effect, even though it lost association with PCF11. So that is something that we are still trying to kind of tease out as to why we're seeing exacerbated repression of PCF11 function, even though it's not associated with PCF11. We think that it could be also leading to mislocalization, which would explain that. But we don't have any new data quite yet to pinpoint what the difference is. But the phenotypes of the cells are different, and the phenotypes of the mice are different.
Right, because she follows up by saying, you know, RNA processing defects are likely to be pleiotropic. Why do you think there is a predilection for neurons, specifically motor neurons?
So we know that mislocalization is different in different cell types, and I don't know if that's one reason that we could be explaining these differences. And also the genes that are expressed in different cell types are very different. So we predict that a lot of the genes that are being affected tend to be these very large genes that encode for proteins that are important in neurons, and maybe that's where the specificity is coming in. The mice also do seem to have kind of a muscle phenotype, which hasn't been reported in the patients. And so that would also be consistent with changes in like ion channel function. But there's definitely hasn't been a nice tissue-wide study done to look at, you know, whether this complex has any specificity in motor neurons compared to other cell types. So we don't know yet.
Great. Sung is asking, as you discussed early in your talk about how there are a variety of subpopulations of neurons, would you say your induced neurons, so I think your early model, do they closely mirror cerebellar Purkinje neurons or other types of neurons?
I mean, it's thought that they are more similar to a cortical brain neuron than the motor neurons. They definitely don't express the motor neuron markers. We used the protocol that was developed in the FOO lab at UCSD at the time. We know that these are not very good neurons. Like they're quick and dirty, and they kind of express neuronal genes and things, but they're not ideal. So we want to also, especially if we're going to move towards studying Alzheimer's disease, we want to move into like better neuronal models and definitely use some of the mouse models that we've created to kind of understand what's going on in real neurons.
Right. And I think just for me to chime in, I think in terms of modeling neurons from stem cells, it's still harder to get cerebellar Purkinje cells. Those protocols are not very well defined, I don't think.
I think the efficiency is like 5% or something. It's terrible.
Paul is asking, from your CLIP1 RNA-seq analysis, do you identify alternative polyadenylation events in any of the other genes causing pontocerebellar hypoplasia?
Yes. Yes, we do. But those genes that we identify the alternative polyadenylation events, typically those patients also do not have motor neuron features. So for instance, like Piccolo was one, and that causes pontocerebellar hypoplasia type 3, which has some ocular involvement, but not motor neuron disease. So whether those are contributing to the phenotype is unclear too.
Okay, well, I have a couple of questions that I will ask while we're waiting. So anyone, if you have more questions, type them into the chat. So I have one kind of just really general question. I think we chatted a little bit about this. So it always strikes me with these RNA binding proteins that are associated with neurodegeneration is they all seem to go into the cytoplasm. So, TDP43, I think FUS does it right now, the CLIP1. So do you think that they're all doing the different things in the cytoplasm, or is that like, how do you relate like CLIP1 with that common cytopathology really?
Yeah. So CLIP1 is a little different than some of these other ones where it doesn't have an intrinsically disordered domain at all. So it's not proposed to be prone to aggregation in general. However, it does not have a nuclear localization signal, so it needs to be brought into the nucleus with something else. And whether that thing could be aggregating with it or leading to like this aggregation is unclear. So I don't know. It was very surprising to us. There's another paper that probably will be coming out shortly, showing that there's also other mutations in CLIP1 that lead to mislocalization. But I mean, a lot of them, there's only one human mutation in CLIP1, and everything else is been synthetically generated, I guess, in the lab, right? So we don't have a lot of information about how changes affect localization and function beyond what's been kind of done in yeast. And there is like functional divergence. So the yeast CLIP1 has no kinase activity, and the human CLIP1 cannot compensate for it. So there's still like a lot that's unknown about, the protein itself, and then if it is related to any of these other types of RNA binding proteins in their mechanism of disease, like the gain of function is similar or different. I have no idea, but I'm excited to find out.
Yeah, no, I just thought that was a really striking finding. I'm going to ask one more quick question, and then I'm going to ask one more question from Ellen. So have you looked in any of the published AD RNA-seq data sets if there's upregulation of CLIP1 or its targets, and just the work that's out there so far?
Yeah, so there's not a good correlation between the transcript expression of CLIP1 and the protein expression of CLIP1. However, in that one paper I highlighted where they did look at APA and CLIP1 overexpression in AD brains in that subset of 36 patients, that was at the transcript level, and it was only increased by 50%, so 1.5x increase. And then from some single cell RNA data sets from Alzheimer's patients, it's been shown, I think, that the cell type that has the increase in CLIP1 expression are the excitatory neurons and not the other other cells of the brain at this point. So
do you see upregulation of those targets though, those long mRNAs?
We've never looked, to be honest, but that's a great idea to look to see if those are increased as well.
Okay, Ellen has another question. So she's asking, when you look at the expression in your AD tissue and you see altered expression in the late stage, the high Brock stages of AD, but not the earlier stages, the low and middle Brock stages, how do you interpret those results? Does this imply poor power in early stage AD, or that things just go to pot in late stage AD?
I don't know. So we were only this was just like one pilot test with it's from our Alzheimer's Disease Center, and Zhangwei Zhu just selected 10 slides, and here's 2 patients, one has an infarct control, here's a non-AD control, here's a mild, moderate, he just gave me those slides, and then we stained them, and it just happened that all of the 3 that he gave us had high expression of CLIP1. I can tell you there was another one that had very high expression CLIP1, and that patient did not have dementia, but it was like a 101-year-old individual. So it was one of these super old aged people who there was no cognitive phenotype, but they also had pretty high expression of CLIP1 and also some mislocalization, but they didn't have any dementia. So whether it's just something that happens, the other ones were age matched. So the ones I showed were age matched, but whether this also happens naturally in the population and there's some mechanism to counter that, I don't know. We have now the mutant mice. So when we made our CRISPR mice, we also got knockout mice. And so we wanted to, look to see if we could reduce the level of CLIP1 in these mice and see if we could change any of the pathology or behavior that exists in the AD mice models, but we don't know yet. But it's something we're very interested in. So if anyone is more of an AD expert than me, which wouldn't be hard, and is interested, please let me know. I'm happy to work together with people.
I mean, it definitely sounds like, there's some good precedent to do a larger study with more patient brains. Yeah, especially maybe in those early, because she also says even a little change in splicing can have a huge effect. So a 50% change in expression could actually be a lot.
Yeah, especially if it's not, like the transcript is not well correlated. So it could lead to huge changes, especially if it's aggregating and accumulating elsewhere.
Yeah.
Right.
Great. That brings us to the end of the questions so far in the chat. Does anyone, I think we have like 1 more minute. Does anyone want to chime in with any other thoughts?
Just one little comment is that we find a lot of hnRNAs, very long transcripts in aggregates. That is, the encoded proteins are in aggregates. And so that's one way of getting something out of circulation, is to sequester it in aggregates. Mm-hmm. That might be part of what you're seeing.
Okay. So you would just look for co-expression of stress granule markers, essentially? Is that how you determine that it's aggregating?
Well, no, we actually isolate aggregates.
You do, okay.
Yeah.
Well, great.
Well, thank you. Let's thank Ashleigh again virtually for a great talk. Thank you very much.
Thank you for all the wonderful questions.
Before Thanksgiving, yeah. And I definitely look forward to talking more, and hopefully people will stay in touch if they have additional questions.
Yeah, my email, I think, was on the first slide, but I'm, easy to find online. And if there's anything that we could work together for, just please let me know. I'm happy to collaborate with people.
Great.
Thank you.
Thank you. Bye, everyone.
Have a good holiday. Bye.